Welcome, everybody. Good evening. Welcome to the 7th Annual Midsummer Nights Science Lecture Series. This is the second lecture of our summer, exploring the genome's dark matter, what the frontiers of genomic research are revealing about cancer. I see some familiar faces here, so I'm just wondering, I love to give Levi a sense of his audience. How many of you were here last week for... Oh, so I guess you enjoyed last week. The expectations are high, Levi. That was an amazing session on the future of therapeutics, road to vital therapy, and I expect today will also be enlivening. As you know, Midsummer Nights Science is an annual lecture series that explores key advances in genomics and medicine. We hold it each summer, and it's free and open to the general public. So that's students of all ages, and I know we have high school students in the audience and undergraduates, our neighbors, and with our Kendall Square colleagues, really anyone who's interested or curious about science. So welcome. I hope you find the evening really interesting and that you'll come back for other events in the series and for future public events that we'll be holding in the months and years ahead. As I mentioned last week, for those of you who can't join us in person, we do record these events from these remarks through the questions, and they'll be available on our website. Last week's session is already available and can be seen on YouTube. And we also have people in the audience who are tweeting the event. For those of you who would like to tweet the event as it happens, the hashtag is BroadTalks. Okay. So we're excited about tonight's topic and tonight's speaker, Levi Garraway. You do have his bio in the sheet in front of you, so I won't go through it in a lot of detail, but I will mention that he's been affiliated with the Broad, more or less, since its founding, although he mentioned to me this morning at that time he was a postdoc in the lab of someone who was affiliated with the Broad. You'll also notice he's a product of Harvard. He went to Harvard College, getting his undergraduate degree there, and then over to the medical school where he got his MD and PhD. He's currently an associate professor of medicine at Harvard Medical School and the Dana-Farber Cancer Institute, and a senior associate member of the Broad Institute, where he leads several large cancer genome analyses and functional genomics efforts in the Broad Institute Cancer Program. We'll have lots of time for questions, I hope, but for now, let's not delay any further, and please join me in welcoming Levi Garraway.
Well, thank you very much, Vivian, for that kind introduction, and thank you guys for coming out on a summer evening, on a beautiful summer evening. There's lots of other things you could be doing than aside from listening to me, and I guess if next week it's only a fourth full. I know I failed miserably, but hopefully that won't happen. What I'd like to do in the next 35, 40 minutes or so is to give you an overview of how we are in the field of cancer, are thinking about cancer, and really kind of from the ground up, understanding the underpinnings of cancer and how we can use that to not only improve biological knowledge, but also increasingly build more rational therapies, and understanding the genome is integral to that, and hopefully you'll see some examples of why that is over the course of the talk. But I'll begin with pointing to a book that many of you may be familiar with. This is The Emperor of All Maladies, which was written by Sid Mukherjee. I can call him Sid because he and I were residents together over at MGH, and in fact, my wife and I were in the same childbirthing class as Sid and his wife. So, but through all that, none of us had any idea that he was writing this book all along during that entire decade, which eventually won the Pulitzer Prize because it was a very eloquent biography of cancer, as he likes to describe it. And one of the many vignettes that points to some of both the challenges and the triumphs that we in the field of cancer and patients who have struggled with cancer have experienced over the past century or so was described in his depiction of the sojourn of Sidney Farber, who was a pathologist working at the Brigham and Women's Hospital and Children's Hospital at the time, back in the 1940s, and how he came across a molecule called aminopterin, which really still, the work that resulted from that still stands as one of the great triumphs in the face of what, of course, is such a horrendous disease. And the reason why it was a triumph is because this drug led to the first instance, and this was a paper that he published in the New England Journal of Medicine in 1948, the first instance where a medicine was used to achieve remission of advanced cancer. Before that, kids with leukemia were admitted to the hospital, but more or less they were admitted there to be studied and to progress through cancer and to die. But this was the first example, not only that you could achieve remission, but that you could achieve remission with a molecule, with a chemical compound injected as a medicine into patients. And with the benefit of hindsight, one can look at sort of some of the principles of how this event came to pass, and there are lessons to be learned about how we ought to think about cancer now. So one of the lessons was that Sidney Farber, when he gave this drug, he wasn't just sort of rolling the dice, he actually had a hypothesis. His hypothesis was that leukemia cells might have an unusual dependency or unusual reliance on folate metabolism. So many of you know folates are in lettuce and leafy vegetables, et cetera. And there had been a series of reasons why he thought that, but one of which was that in some cases, kids who received folic acid or folate compounds sometimes saw their leukemia grow faster. So he thought, well, maybe the leukemia cells are depending on folate metabolism. And it happened that at the time, he was able to get his hand on compounds that inhibited folate metabolism. And so essentially, although he didn't use the term, this is a term we use now, he used a rational therapeutic that was deployed in a patient based on a hypothesis about the biology of the disease. Now, this all seems very sensible. I mean, it sort of seems like that's what doctors should do. You should understand the biology of disease, and you should give a drug that will attack that biology, and patients will get better. But in fact, I hate to say it, but we don't really get to do that in cancer as much as we would like. We are usually trying to carpet bomb cancer with drugs that are generally toxic to cells, and we kind of hope that those drugs will be more toxic to cancer cells than they are to non-cancer cells. And sometimes that's true, but all too often it's not true. And especially when we're talking about metastatic cancer, advanced cancer, it's still all too often not true. But when we look back, this is in essence what was done, and it led to dramatic responses. But unfortunately, those responses were transient. And of course, we still wrestle with this, particularly when we use a single drug to try to treat cancer. We may be fortunate and be able to achieve a remission or a response for a time, but invariably what happens is that the tumor will progress. And so understanding mechanisms of resistance, and one of the other great triumphs that occurred over the past century against cancer happened in the 1960s when other investigators applied therapeutic combinations that were thoughtfully implemented for the first time. And really it was that led to cures of at least several types of leukemia, lymphoma, other types of cancers. And so really, if we sort of think about this path that I've now laid out for you schematically, this is the path that we would like to be on, and in fact can be on in the era where our biological understanding is so much more vast. And so what we can often do is think about different cancer types and ask, where are we on this path? In some cases, we've gotten here. We understand the biology well enough, and we have drugs that will achieve tumor responses, but we're now struggling with resistance. In other cases, we are already here. In other cases, we're still over here. Our biological understanding is still nascent. But we want to be on this path, and advances over the past several decades have told us that we can be on the path. So there is reason to hope, even though it is a challenging and sometimes desperate condition to be diagnosed with cancer. So this is all interesting, but where does genomics come in? Well, genomics essentially over the past 10 to 20 years has taught us a great deal about tumor dependencies, or what it is, what biology it is that cancer cells tend to rely upon, sometimes unusually or specially, and that's the beginnings of how we start to move down the path. And the reason is that cancer really can be thought of as a genomic disease, as a disease of the genome, because in essence, cancer cells, if you compare cancer cells from an individual to non-cancer cells of that same individual, you will inevitably find changes in the DNA or mutations in the DNA. And essentially what we do in the field of cancer genomics is systematically study those changes, identify what they are, and then come up with hypotheses about why it is that some subset of those changes causes that cell to become cancerous. So why is it now that cell invades surrounding tissue when it shouldn't? Why is it that it doesn't die when it should? Why does it spread to distant sites and keep growing? The answers are often resident in not just the genes, but in the types of mutations that we see in genes. And so in this case, in some ways, a picture can be worth 1,000 words. I understand that there are many high school students in the audience, and so maybe by now some of you have already seen pictures like this. These are the chromosomes in which the DNA of each cell resides, and there are 23 chromosomes, 22 what we call autosomes, and then there are the 2 sex chromosomes. This is a male because of an X and a Y, and then a woman would have 2 X chromosomes. So this is very nice, tidy. You can take cells and do a karyotype analysis like this, and most normal cells look quite beautiful, in fact. But in cancer, the situation is very different. So you can even see kind of at a global level, and this is color-coded for effect, because what you can really see is that it's almost like a bazooka was taken to the genome, and it was blown apart, and it was sort of stitched back together in a haphazard way. And there are all types of derangements, and if one zooms in down to the specific molecular details, one sees even more alterations. So cancer genomes are deranged, and that is true for nearly all cancer genomes, and the essence of this derangement, or subsets of that, are really germane to why the cancer cell is still alive, why it invades, etc. So just as a very brief reminder, I'm sure many of you in the audience will know this very well, but in essence, just to remind you, the DNA consists of genes, and then there's a lot of the other DNA, which I'll get to in a minute, but just for the business portion of the genome consists of genes, which of course give rise in the cell to proteins, and that process usually works extremely well, but if it happens through normal processes of cell division, or sometimes because of exposure to ultraviolet light or tobacco or extrinsic carcinogens, if it happens that certain regions of the genome or genes become mutated or damaged, they will generate incorrect proteins and disease will occur. And so if genes that are involved in cell growth and proteins, therefore, that are involved in cell growth are mutated, then the result of that becomes cancer. So in essence, this is now the revolution in our understanding over the past several decades is really to think about cancer from the ground up, the ground being the genome, and how the genome becomes deranged, and therefore what proteins become altered. And really, the rational therapeutics are about directing therapeutics against those altered proteins or the altered cellular biology that results from those damaged proteins. Now, one of the major revolutions, and actually I would say this is really one of the transformative events in the past decade for those of us who study genomes, has been in the area of technology, and in particular there's a technology that is called massively parallel sequencing, used widely at the Broad, as you might imagine, but now at many, many other sites around the world, which have made it possible to sequence an entire human genome, and actually many times over in a short period of time. So you may recall that it took many years to generate the first human genome sequence. Well now, if you think about the human genome is 3 billion base pairs. The base pairs are sort of the basic letters that make up the genetic code. There are 3 billion of them in each human genome. Well, one of these instruments, and it's not even now one of the most updated instruments, can generate 40 billion base pairs per day. So, on the order of 13 or 14 human genomes can be sequenced per day. And that's extremely powerful because now we don't have to wonder if we want to know what happened to a cancer compared to the normal DNA of an individual. We don't have to wonder. We can know in a matter of days exactly what happened. And that knowledge and the ability to know that across many, many types of cancer has been transformative in our understanding of cancer biology. So coming back to the first phase of this critical path, I described to you the concept of tumor dependency, some biological process on which a cell relies unusually, a cancer cell relies unusually. Well, the genetics and the ability to sequence genomes at large scale have now accelerated our understanding of tumor dependencies, because essentially if it's locked into the genome, if the mutation is locked in, essentially what that means is evolution has taught us that might be important. And so we have been able to expand our understanding of important cancer genes and important biological processes greatly over the past several years. Concomitantly, the pharmaceutical industry has responded and has begun to engineer a whole fleet of chemical matter and brought those into patients in clinical trials that allow us to begin to say, well, maybe we can target some of these deranged pathways in a smart way. And there are a variety of examples where when that has been done successfully, there have been major responses in cases where the patients would have had no options otherwise. Now patients can live for many months or even many years on some of these targeted therapeutics. So clearly this path is working, and understanding the genome is integral to the fact that that path is working. Now, we're not done. We have not cured everybody, but we are clearly on the right path based on this knowledge. Okay, so what I'd like to do is just give you a couple of specific vignettes about that hopefully will give you a flavor of the kinds of unexpected knowledge that can come from studying cancer genomes and where it might be taking us. So I'll give you a couple of vignettes that are insights into cancer biology, and then I'll close by some insights into what we call personalized cancer medicine, which is really still more aspirational than real, but we are very rapidly moving down that path, and it's a very exciting time to see how the basic genetic understanding is moving ever more rapidly into the clinical arena. All right, so you may be wondering, or maybe now you've decided, well, why is the title of this talk called The Dark Matter? Well, the short answer is that maybe I was too slow to give a real title, and so Vivian and others said, well, let me read one of his papers and come up with a title. But it turned out they did a good job. So, but now, dark matter, for those of you who are not physicists, the real term, I mean, it's kind of a cool term, and we use it in cancer, but it's not really quite accurate. Dark matter refers to, in the universe, it turns out there's actual physical matter, but the actual physical matter only accounts for a minority of the total matter that seems to be there, but based on the way physicists calculate mass, et cetera, based on particles and signals from the outside around the universe. So there's this unmeasured matter called dark matter, which must be there. It's inferred that it must be there, but we can't really study. We can't access it, and so we don't. It's unsearchable right now, but it must be there. And a large part of the genome really can be thought of in terms of dark matter, because there are large swaths of the genome, which they obviously exist because we can sequence it, but we really can't interpret it all that well. And I don't have time to go into all the details about where that dark matter sits, but one very simplistic way to look at it is that areas located in between genes. So it turns out that only about 2% of the genome gives rise to genes, the part that encodes proteins that make up the machinery of the cell. The rest of the genome contains a lot of other regions that are important, but many of those regions are difficult to really understand. And yet we've known for a long time that those regions must be important. And there's been a lot of work in basic genomics that has sorted out a lot of how that is important. But just to be very simple, one obvious sort of area that made us curious was that in most of the studies, until very recently, most of the studies in cancer genome, understandably, focused on the genes, the region, the subset of the regions that actually give rise to the gene, that encode genes and therefore give rise to proteins. And so, but one of the other regions in between genes that we understand reasonably well is what's called the promoter region. So you can think about this in terms of if the gene is sort of the light bulb itself, then the promoter region is the dimmer switch. And obviously, this dimmer switch, in some ways you can turn it on or off sort of discreetly, or you can dial up and down the intensity. And that's what promoter regions basically do. They're very complicated. There are regions that draw in proteins and various other factors to dial up and down. And almost every gene has a promoter region, and then there are other regions that modify that further. But these promoter regions, subsets of them can be thought of as part of the dark matter because we don't always understand exactly how regulation occurs and how does that change from cell to cell, et cetera. But you can argue that, well, just searching the genes, the genes proper for mutations is a little bit, maybe that's narrowing our understanding artificially. And you could even say, well, maybe this is kind of like the proverbial drunk looking for his keys under the lamppost. I mean, it's sort of like, well, we look there because we sort of understand how to look there, but maybe the keys are over here somewhere. So the question, a very simple question, that two members of my lab asked about a year or two ago were, are there mutations in the dark matter of the genome that cause cancer? And so actually, Iran Hodis, who is a graduate student or an MD PhD student now, and is also a computational biologist, sort of asked the question in the very simplest sense, which was just to ask, are there any base pairs, any letters anywhere in the genomes of melanomas that we've been sequencing that land outside of the gene proper, where there are mutations that are piling up in melanoma? I mean, it's a very simple question. And we actually did not really believe that the answer would be yes. And I won't go into why, but it just sort of didn't really seem all that plausible, because it's much easier to make a cell deranged by mutating a protein, seemingly, than it would be by mutating something else in the genome that doesn't even regulate, that doesn't even change the function of proteins. But sure enough, the result of that analysis, and then Franklin, the other person I showed, led to the follow-up, revealed that in melanomas, there are two mutations that land in the promoter, and it wasn't just that there are two mutations that land in the promoter, it was that 70% of all melanomas had one or the other of these mutations. This result was so surprising that we didn't believe it at first, or at least I didn't believe it at first. And in fact, it turned out to be a little bit challenging to prove that this was really true, but I won't get into all why, but once we finally realized it was true, it was shocking, because now the frequency of these mutations in melanoma was even more common than some of the best known mutations that we already have therapies against. I mean, by far, it was more common than some of the other mutations that we had been studying for 10, 15, 20 years in melanoma. So it just didn't seem at first plausible that a region in the sort of in-between section of the genome would actually do this, but sure enough. Now, Franklin came in and then did an experiment to not only show that, yes, those mutations were there, but they were actually doing something. And so the experiment that he did was very simple. It's essentially put the mutations into sort of an artificial system where you can see, well, if you have the mutation in the promoter region or not, does it change the expression of a gene? So in other words, you put the mutation in, you ask, does the intensity of the gene go up? And the result of that, or part of the results, are shown here. So this is sort of the normal situation, and when you put one or the other mutation in, boom, yes, indeed, they go up. And he did this in several different cell lines, 3 of which are shown here, and boom, they always go up, and they always go up significantly. So the presence of one or the other of the mutation led to increased expression of this gene. The gene is called telomerase. Actually, for those of you who want to go Google it later, telomerase is a gene that it turns out that the chromosomes, if you divide your cell, you need to actually, every time you divide a cell, you have to repair the ends, the very ends of the chromosomes. Otherwise, they get ratty, and eventually the chromosomes will break apart. So cells have to repair the ends of chromosomes. And in fact, cancer cells in particular have to repair the ends of chromosomes, because if they don't, they cannot be immortal. So almost every cancer cell has to somehow dysregulate telomerase so that the ends of its chromosomes can always be repaired every single time it divides, and it can live forever. So this is actually a very important cancer gene, but we really hadn't understood how its dysregulation in cancer occurred, but certainly in melanoma, it looked like the answer in the vast majority of cases was these promoter mutations. Now, shortly after our paper was published, there was a group from Johns Hopkins that did a survey. They had 1,300 or so tumors of different types lying around, and they were able to very quickly take the knowledge of the mutations that we published and sample their 1,300 tumors. And lo and behold, a large fraction have a very high frequency of, again, one or the other of these mutations. And so, for example, some brain tumors have more than 80% of those brain tumors have one or the other of those mutations. And again, bladder cancer, liver cancer, oral cavity cancers, a large fraction of tumors have a high frequency. So it turns out that, and who would have thought, but it turns out that mutations in the promoter region of the telomerase gene are now considered among the most common base mutations or point mutations in all of cancer. So who would have thought that the most common mutations in cancer are not in the protein coding section, but they're in the dark matter, if you will. And so now, but this gene, or the protein that is regulated, can actually be targeted by small molecules. So there are now renewed efforts, whereas there had been efforts had kind of waned historically about whether it would be really interesting to target this molecule. And I think now there are brand new efforts, including potentially some activity at the Broad around developing therapeutics against telomerase, largely as a result of this observation. Because clearly now, not only do we know that it's specifically dysregulated, but we can also even imagine, well, these are the types of cancers where we would deploy that drug first because they have a genetic alteration that is dysregulating telomerase. So this was quite an unexpected but an exciting result, and it was exciting enough to get published in the New York Times, which actually I have to say I was quite surprised about. I didn't think that this must have been a slow week because I would not have thought, with the fiscal cliff and sequester that, but anyway, it was kind of fun for that. Okay, so the other vignette that I want to just tell you briefly is also related to the dark matter, but now this kind of shifts gears and talks about the evolution of cancer, and I'll tell you why the evolution of cancer is important for a moment. Now, for those of you who have taken high school or college biology, you will know that the sort of classical Darwinian view of evolution is one of gradualism, which can be depicted like this. If this is time, now we're talking about cancer evolution, not species evolution, but the concept is basically the same. So if we talk about the number of alterations per cell, every cell division of a cell over the lifetime of an individual, there are some mutations that are accrued, but in some subset of cells and a subset of people, they get they get unlucky enough to accumulate the wrong subset of mutations, and at some point cancer arises. Now, this process happens stochastically in every cell, but there can be certain things that accelerate it. So for example, if somebody smokes regularly, now the slope of this may go up. So the likelihood of developing just the wrong collection of mutations shifts, and that means that if this is your lifetime, your chances of getting cancer go up. Another way in which you can shift this curve is by having an insult that occurred at some point in your life. So for example, if you were spending a lot of time outside in the sun and you got blistering sunburns as a child, you may now have dialed up the risk in your skin cells, or what we call the melanocytes, of getting melanoma. So this is another way you can increase the sort of pace of cancer. But basically all of this falls under the realm of what we call gradualist, Darwinistic type tumor evolution. And in fact, the vast majority of tumors will arise this way. But we had an observation, and I'm not going to walk through this slide, but this was the figure from our paper that said, strange things can happen to cancer genomes. There can be these really complicated, dizzying rearrangements of the genome, and we described one of them. This is the scientific rendition. This is the artistic rendition. So hopefully that clarifies, for you now what's going on. But what you can see by the color coding is that what the end result is that different regions of the genome on different chromosomes can get stitched together in a very strange and fascinating way. Now, we wanted to understand this process a little bit better, and because of time, I can't get into what we think, the basic cellular derangements that lead to this, although I think there's some very interesting work there. But it turned out that another MD PhD student in my lab, by the way, one good thing about working at Harvard is you get really, really smart people who just wander into your lab and they want to do experiments. And I told you about, Iran, who found, these telomerase mutations just by kind of futzing around on the computer one afternoon. And then there's another computational biologist, MD PhD student, his name is Sylvan Baca, who initially came into the lab and he was doing some experiments at the bench and studying proteins. But when he started learning about cancer genomes and stuff, it turned out that he was a math whiz, and he looked at these rearrangements. He said, "Oh, we can sort of sort out this. This is just a graph theory problem. We just need to, treat the, the breakpoints as nodes and and I was like, "Oh yeah, that's what I was thinking." Yeah. So he went to work and developed applied graph theory and developed an algorithm to describe these events. And it turned out, again, totally unexpected, that these chained events are incredibly common, particularly in prostate cancer. This is one of the areas of cancer that we work on a lot in the lab. So common that we thought it was worth a name because people tend to name these things. And so Sylvan came up with a really elegant name, chromoplexy. plexy is Greek for weaving or braiding, and so it seems like a really appropriate description. And this is a depiction of, we sometimes show cancer genomes as a wheel, where the chromosome 1 is here all the way through all the chromosomes, and then the X and Y are here. And you can see the spokes on the wheel are where the rearrangements have occurred. So this is one way to visualize them. And then they're color-coded based on one or another chromoplexy events. But what was striking here was that almost all prostate cancers that we looked at had at least one of these events, and 2/3 of them had at least two. And then in some cases, we could show that there was a temporal order, meaning one chain arose, and then later on another one arose. So we could basically learn a lot about sort of the temporal pattern of how they arose. And then the most important observation was that these chains would commonly affect genes that we know to be involved in cancer based on prior studies. So all of these together gave us a window into, I just told you about sort of evolutionary gradualism in cancer, but it turns out that this phenomenon told us that there's another evolutionary process going on, which is much more akin to punctuated equilibrium. So again, those of you who have taken college biology will remember that the concept of punctuated equilibrium was introduced by Stephen Jay Gould because he didn't like the idea that gradualism didn't explain the fossil record. The fossil record didn't have intermediate species, it had discrete species. So he thought, well, maybe there are these punctuated events followed by relative stasis, and this is how you get species evolution. Well, what it looks uncannily like is that these chromoplexy events are these punctuated events. There's one that happens, and then another that happens, and another happens. And these may be an alternative way or a complementary modality compared to gradualism for how tumor evolution arises. And so the recognition of this rearrangement pattern allowed us to discover that there may be a companion model. Just like there's gradualism, there's punctuated evolution, and this, at least in prostate cancer, and probably in other cancers as well, this is how cancers arise. Now, why do we care? Well, there's a couple reasons why we care. One is if we can understand the process of how these events occur, and if we could slow them down just a little bit, that could be a cancer prevention therapeutic. To prevent cancer, you don't have to eradicate the process. You just have to slow it down long enough so that it doesn't cause problems during the lifetime of an individual. So we didn't know about these before a couple of years ago, but now that we know about them, can we understand why they occur? Why do they occur more commonly? Is it really true that they're more common in prostate cancer than others? And can we just slow them down just a little bit? And would that be a way to prevent prostate cancer? Cool idea. I mean, if we hadn't done genomics, we wouldn't know about it. The other reason why it could be interesting, and this is a little bit even more speculative, but since we know that these events tend to involve cancer genes, it might be that if we found that a cancer gene was altered and it happened to be altered in the context of one of these events, maybe that means it's more likely that for that patient, that's the important gene. But we'll have to explore that more going forward. So those are two vignettes about biological insights that come from sequencing the cancer genome and avenues into how cancers arise and how, therefore, they might be treated that we might not have thought about otherwise. But what I started this talk sort of pointing out is that it's great to understand the biology, but what we really want to do is to leverage genomic understanding to drive more effective cancer medicine. So for the last few minutes, I'd just like to give you, again, just some highlights about how cancer genomics is empowering that direction in some very exciting ways. So the concept here, again, it's very simple. What you want to do is when patients come to the hospital, not only do you obtain a biopsy and make a diagnosis by looking under the microscope or doing x-rays, you also will now extract DNA. And by doing that, you will sequence their genome or a portion of their genome, and you'll get a list that looks something like this. On the one hand, you have a list of mutated genes, and on the other hand, you have a list of drugs. And if you find, something like this, where there's a particular mutation that matches up to a particular drug, then the doctor says, okay, fine, I'll give this drug. And you actually want to do that, even if this would be an unusual event in that particular cancer. So let's say you had a patient with pancreatic cancer, and they came into the doctor's office and they sequenced the genome, and it turned out that they had a c-KIT mutation, which in other cancer types can give dramatic responses to a drug called imatinib. Well, would you rather try imatinib if your pancreatic cancer had a c-KIT mutation, or would you rather try gemcitabine, which improves survival by 3 or 4 weeks? You be the judge. So that's the concept. And again, this is one of the things, well, this seems obvious. Well, we should just be doing this. But in fact, we haven't been doing this because it hasn't been possible until very recently. So there are 3 fundamental principles that make this possible, at least in theory. One is that molecular pathways involved in tumor survival and progression are very often activated by genetic alteration. So I've just shown that what this slide shows is either by pie chart form or bar graph form of several common solid tumors: lung cancer, colon cancer, melanoma, brain cancer, ovarian cancer. And we're just tallying up, and this is not all the genes that are mutated, this is just the genes where you can argue there might be, knowledge of these genes might change what you would do clinically. Maybe you would use a different FDA-approved drug. More commonly, you would enroll a patient on a clinical trial with a particular agent. In some cases, it tells you what drugs not to use. But the sum total of this is that, at least in the several major tumor types and others that I haven't shown you, 40 to 60% harbor at least one genomic alteration that affects what we call an actionable cell growth or survival mechanism, meaning that if you knew about that mutation, you might think differently about how to manage a patient or what clinical trials to enroll them on. So that's an important principle. The other one, the second one, is that anti-cancer agents that target many of these cancer pathways have entered clinical trials. Now this is the first time in history. So what I'm showing you here, this is a schematic diagram. Here's the cell membrane, and here's a bunch of proteins that sit in the membrane. They send signals down these pathways. Those pathways then change gene expression patterns. The point about this, though, is that every shape here is a different protein, and every shape that has a solid line around it has at least one drug, a targeted drug, in clinical trials or in FDA-approved status directed against that protein. And these are proteins that are mutated or pathways that are altered in cancer. So this is the first time in history where we've not only had the genomic understanding of cancer, but also the ability to a large repertoire of targeted agents in clinical trials, unprecedented repertoire of targeted agents in clinical trials for the first time ever. And the third point is, which I've just been telling you, we now have the technology. The genomic technologies enable tumor genomic profiling, not just to discover new dark matter events or new genetic alterations, but to identify them in the clinical arena. So this is a graph that we show a lot, which is, so Moore's law, you may know, is the rate of decrease in cost of data processing because of advances in the semiconductor industry. This is why we can, our iPhones work well, is because of Moore's law, and this is a log scale. The cost of data processing has fallen more or less along this slope for the past couple of decades. Well, this is the Moore's law equivalent for the cost of sequencing a cancer genome. So, if we sort of think about how much did it cost to sequence a genome in 2001, it was $100 million, and then now we're all the way down to the point where it's well under 10,000. Actually, it's now down into the low thousand range. So the cost of sequencing a genome, the rate of decrease has rapidly outpaced Moore's law. So we have the technology. We can do this, and it doesn't cost all that much money to do this. Now, it doesn't mean, again, this sounds easy, but it's not all that easy. And one of the reasons why this is not easy is that if you think about what the outcome of this is, if you think about data points per patient. So as a doctor, for the typical patient who comes to the doctor's office or goes to the hospital, you take data points. You take the history, you take the physical, you get labs, you get x-rays, you maybe send some tissue for pathology. And that gives you on the order of dozens of data points per patient that the typical doc aggregates, and they write their assessment and plan. They give you a script, and they send you home and say, come back in 2 weeks. Okay, now if you start adding even the most rudimentary mutational profiling approaches, one of which our lab developed and is in use at Dana-Farber, now you get up to the high dozens, maybe 100 data points per patient. But once you start saying we're going to sequence the protein coding region of the genome or the entire genome, well, now you're way up to hundreds of thousands of data points, and you now have vastly exceeded even smart Harvard doctors' ability to integrate data and make an assessment for patients. So what that means is, okay, this is coming, but we now need the help of tools computational tools to help us sift through the data and make sense of it? Well, once again, thankfully with the Broad Institute, aside from all these sequencers, they have lots of compute space, and they have really smart computational biologists who help figure out what tools and methods to use. One of the really smart computational biologists, who also, it just what happens, is a medical oncologist, so it's like the perfect convergence of skills, has developed an algorithm, and there's a lot of big words, precision heuristics for interpreting the alteration landscape, and you have to have an acronym. And so the algorithm takes all the mutational output of genomes and essentially comes up with a rule-based approach to say, all right, I'm going to rank them based on the possibility that you could act on these clinically. And if you couldn't, well, maybe there's some biological knowledge that might be useful, and they're basically ranked accordingly. Now, for the nerds in the audience, you may realize that the acronym PHIAL comes from the Lord of the Rings, so this is the Phial of Galadriel. Yes, all right. So she gave it to Frodo with the quote, "May it be a light to you in dark places when all other lights go out." So we nerds felt, it gave us goosebumps. This has to be the acronym, because this is what we're doing for patients. They have no other options. They have failed cytotoxic chemotherapy. They have metastatic cancer. We got to give them some glimpse of what could we try next. So, and so this is the output. So this is one of the first patients that we subjected to tumor genomic profiling. This patient had widespread melanoma. We did tumor genomic profiling, found that they had a mutation in a gene called BRAF, which allowed them at the time to go on a clinical trial of a RAF inhibitor, and the result of that trial was this. So this right here is what we are trying to do. Now, I hate to end on a bad note, but this was great, but it only lasted a little while. A few months later, this is what he looked like, and what this underscores is the challenge of drug resistance. So one of the other areas that we are applying genomics to understand is not just to characterize the genome here and say what drugs should we give, but to characterize the genome later and say, why did the patient become resistant? What does that teach us about what drugs we should give next? And not only that, what combinations we need to give to avoid the problem of resistance. The good news, though, is that in many cases we've done this. We've achieved impressive responses. Yes, they are transient, but what that means is that understanding resistance and overcoming resistance is the final path to cure or long-term cure. We want to be over here, durable control. So we are putting aggressive efforts into sequencing cancers, not just over here, but over here. So this is a schema. And actually, this is now arguably sort of a new investigative realm for cancer biologists and clinicians, where patients who come through the door, so now the idea is, the patients are our partners. We're going to treat them, but we are also going to enroll them in trials. We're going to try to have ways in which we can sequence their tumors and identify molecular alterations. Often that may involve a fresh biopsy when they come in or if they progress on a certain therapy. Now, obviously, there's genomic profiling. As I mentioned, there are data interpretation algorithms. All of these are still very much in development, but as you can see, the tools, the components are in place. So now, with that, oncologists can make a decision that takes into account not only the H&E, the history of physical labs, but also potentially genetic or other molecular information. You may, if a patient's on a clinical trial, take another biopsy there because you need to understand when you gave the drug, did the drug do what you wanted it to do? Did it actually hit the target that you were giving it against? If they get a response and progress, you now may do another biopsy to understand why did they become resistant? Does that tell you about a salvage therapy they could give, or does that tell you about a new therapy that you're going to give the next patient together, perhaps in combination with the first, to try to prevent the problem? So we are not done, but we are in an era where the ingredients to make transformative advances have never been there before, and they are working. We have examples. Everybody who takes care of patients has examples of patients who have been on one of these targeted agents and had major responses. So we know that we are on the right track. A long way to go, but it's a very exciting time. So with that, I'll stop. I think I'm almost on time. And you certainly can appreciate that these efforts take many people, not just people on this slide, but many others who are not on the slide. But it's very exciting to be at the Broad and to be able to bring the Broad to the clinic in a way that hopefully will help many patients with cancer in the future. Thank you.
Thank you, Levi. So I'm holding this phone for a few reasons to remind me to tell you a few things. The first is that we actually have a large number of science writers in the audience, Levi. I didn't want to tell you about it beforehand to make you nervous, but I do want to welcome Science Online Boston to this event and to thank you. I've been watching the Twitter feed for all the comments that you've made. The second is to say that I'm about to wander around with this microphone to anyone who has questions, but there are also two other ways to ask questions, and we will be trying to get to those other questions as well. One is by Twitter, so if you use the hashtag #BroadTalks, some people from communications are monitoring that and will ask questions for you, or you can email to midsummer@broadinstitute.org. And just to keep my promise that I will take a question from a Twitter feed, one of the tweets was about statistics and numbers. So you brought up a large percentage of mutations in melanoma had telomerase mutations, and and the question was like, out of how many, and more generally, what sort of numbers do you need to get at the genes that might be good targets for therapy?
Yeah.
And if you can raise your hand, I'll find another question.
So you want me to answer that one now?
Yeah, answer that while I'm
looking. It's a very good point. So what's behind that question is that for any mutation that's what we call a driver, so it's critical for the biology of that cancer, there are many others that are what we call passengers. They happen to occur in the cell at some point, but they're just coming along for the ride. They aren't really causing the cancer. So the statistical algorithms that distinguish drivers from passengers and developing those has been a major effort at the Broad Institute, and there are a number of people who are the best in the world at the Broad at doing that. But the bottom line is that it's absolutely true to really discover not only the spectrum of mutations, but figure out which ones are the important ones. You do need numbers, and the amount of tumors that you need, it's influenced by a lot of things, but one of which is what we call the background rate of mutation. So some tumors, if you look at them, they have lots and lots of mutations, particularly melanoma and lung cancer, because ultraviolet light induces a mutation damage to the genome. Tobacco induces mutational damage to the genome. So those tumors have a lot of mutations, and most of them are not important to the biology. So you need to look at dozens, even hundreds of tumors to really be confident for which of these mutations are truly drivers. Melanoma, same type of thing. Having hundreds is really what you need to sort of assign kind of the full spectrum of important cancer genes. Now, for the telomerase promoter mutation that I described, it turns out that actually the statistics are a little bit more friendly in that case, because the assessment was out of 3 billion bases, you had to say, what are the chances that the same, that the same tumor of 20 or 30 or 40 tumors would have the exact same mutation in the exact same spot? Now that turns out to be rare. I mean, it's actually simple math. If you say, okay, there are, whatever, in melanoma, let's say there are 10,000 mutations in the genome, but they're diluted by 3 billion, well, so that you can just say, well, at random, they're going to be distributed all over the place, and the chances of it landing in exactly the same spot, even twice, is pretty rare. But so we saw actually the first set of genomes that we looked at, there were about 19 or so, and 17 of the 19 had one or the other of these mutations. That was just astonishingly unlikely. There's just no way. So that's why we didn't believe it. Saying, this can't be right. There's just no way. This could never happen. So in that case, we were helped by just the abundance and the fact that that we had the whole genome to sort of sort through. But in general, sequencing large numbers of tumors is really the only way to get at the full spectrum of drivers because of the statistical challenges.
I wonder if I could ask you to follow up on the prostate example that you gave, that the lifetime risk for men is about 1 in 6. But in many cases, it has no clinical effect during that person's lifetime. The real question in prostate cancer is, when does it go critical or aggressive? And I'm just wondering if that is related to the chromoplexy that you were discovering with prostate cancer.
That's a great question. And just yesterday morning in Sebastian's, I was having coffee with a urology resident who wants to ask exactly that question, because he has a cohort of patients where there are biopsies collected over time, patients who are on watchful waiting, who never developed aggressive prostate cancer, others who did develop aggressive prostate cancer. And so the question is, does the presence of chromoplexy correlate? Because you could imagine that maybe that's the case. If you start seeing these chromoplexy events piling up, maybe that portends the aggressive path, whereas if you never see that, if the genomes are quiet, you won't. And I think there may be some truth to that, but we have to demonstrate it formally, so stay tuned.
Hi, I have a question about just the general process of sequencing tumors.
Okay.
So it's obvious that, well, it's known that tumor populations are very heterogeneous. So I just don't know the method, or how do you account for that much difference in the cells?
So that's a great question. So the question basically is that we usually, the term that we have historically used ever since the early 1970s to describe cancer is clonal, meaning there was one bad cell that was a cancer cell and it gave rise to the entire tumor. But those cells they're clonal, meaning they're the same, they're genetically, quote unquote, identical compared to the rest of the cells in the body in that bad way. But it turns out that cancers are not truly clonal because, as I've been mentioning, every cell division introduces new mutations, most of which are not consequential. Occasionally one will be, but mostly they're not. But the sequencing that I've described to you, not only can it go broad, looking at the entire genome, but it can also go deep, meaning you can sample many individual molecules of DNA from the tumor. And what that means is now you can discover that, whoa, this is not a clonal event. There are many what we call subclones, or lower frequency, meaning of the total DNA that you looked at, only a few % may have this mutation, but it's there. So the answer is, actually, for the first time, we can use this technology to actually go deep and characterize what the term she used was heterogeneity, or the mutational diversity within a tumor. So we can do that, and then the question becomes, well, what does that mean? How do you account? Is that a problem? Is the fact that tumors are heterogeneous, is that a problem for personalized medicine? And some have said, oh yeah, we're doomed. We can never possibly attack all those different mutations in the tumor. Whereas others have said, well, maybe, but the heterogeneity doesn't automatically mean that those different mutations that you test are actually important for the tumor, because let's face it, if a mutation arose during one of the early cell division, like let's say the third cell division, well, it might be present in a quarter of all the tumor, but it could still be an irrelevant, it's just statistical, it could still be an irrelevant event. So the short answer is, we don't really know to what extent heterogeneity is going to be a problem. What I can say, the patient that I showed you had incredible tumor burden, and there was a lot of heterogeneity in that patient, but he had a dramatic response. So the presence of heterogeneity does not automatically preclude dramatic responses, because the founder important mutations may be present in all of the cells. But there are other cases where it will be a problem, and we just have to sort of play it out and see what we... Now, I think the other point I'll say is that there are two ways that you can think of addressing the problem of heterogeneity clinically. One is there may be that in the future we have to come up with higher order combinations, 3 or 4 drug combinations that we figure out how to give patients that will attack most of the essential biology of the cell. The other possibility is that it turns out there have been major advances in harnessing the immune system against cancer. Some of the most exciting results in recent clinical meetings have been that there are drugs that will harness the immune system, and in some cancers, not all, and we don't understand why it's some and not all, you can sometimes get major responses. Actually, dramatic, Lazarus-like effects that are durable. So maybe the immune system will be the answer for how to override. If you can get it to attack the cancer, it'll go nuts and eradicate all those cells, and it doesn't matter about the genetic heterogeneity. So that's one way to think about it.
So my question has to do with the stepwise accumulation of mutations, and the likelihood that some of those could be caused by injurious events. We've also found that certain cancers are initiated or caused by viruses, if not always, but in most cases. I'm wondering to what extent it's been investigated whether such viruses might be those injurious events, and if that's led us to consider whether other viruses may be involved in other injurious events and related to other cancers. In other words, to what extent cancer can be caused by viruses and what those viruses are.
Yeah, so that's a great question, and it's interesting because The idea that cancer was a genomic disease is old, so it's a century old. There was a scientist called Theodore Boveri who looked under the microscope and was able to look at chromosomes, and they were deranged in tumors. So that idea was there, but then it got waylaid by this very influential theory that cancer is, no, it's not about the genome, it's about viruses. And in fact, it's true, because there are clearly viruses that cause cancer. This was true in animal models and chick models of cancer, and in fact, that's what led the Nobel Prize to go to Harold Varmus and Michael Bishop. They discovered that there are cancer-causing genes in viruses, and it turns out they're not from the virus, they're actually from the human, from the host. It's just that the virus grabs it and it mutates it, and then now when it goes back into the host, that host cell becomes a cancer. So clearly viruses can cause cancer. Sometimes it could be because they carry a mutated gene, but more commonly it might be because they land in regions of the gene that in the regions of the genome that turn on normal genes in the wrong way, or in some cases they induce an inflammatory response, and that inflammatory response can sort of, for example, hepatitis, the cancer may be caused in part by the virus, but in part by the inflammatory response that lasts for years and years. So there are several known viruses that cause cancer. The question that you're getting at is, well, maybe some of these genetic alterations can be sort of the footprints of viruses having been there. And I think it's too early to assess the extent to which that's going to be true. However, one of the beautiful aspects of massively parallel sequencing is that you're just sequencing DNA, whether the DNA comes from the tumor or the normal or a virus or a bacteria, it doesn't matter. As long as you know to ask the question, is it there, you can do that. And you can often tell, well, did it land somewhere, and are there characteristics about the regions of the genome where they land? So you can, all of these things are knowable. I just don't know the extent to which anybody has shown that a particular cancer or the sets of alterations might be linked to where viruses come and go in the genome. But it's a great question.
Great. We are getting a number of questions coming in by Twitter and email, so I'm going to do one of those now, and then I'll give the mic to you. Can you give a brief explanation of why drug resistance occurs?
Why does drug resistance occur? Well, there are a few reasons. I'm going to focus the answer on why in targeted therapy, where you get such dramatic responses and we understand why the drug works, why does drug resistance occur? Well, there are three main reasons. Reason number one is that there is a new mutation in the protein that was targeted by the drug that now prevents the drug from binding the protein. It's very simple. You just mutated the protein somewhere else. Now the drug can't bind. The protein goes back to its business of causing cancer, and there's drug resistance. That's one way. Another way is that there's another alteration, sometimes it's another genetic event or something else, that can cause a new pathway to go around that protein that was targeted by the drug. So now it's like if you have a faucet blocked here, you turn the spigot and it goes around, and now you get water coming out of the hose downstream. So if you get a bypass pathway, that's another way that you get resistance. And then the third way would be if you blocked the drug here, but one of the downstream, so if the drug normally does something down here and you turn that on by a different way, sometimes by another mutation, you can then get resistance. So there's sort of three categories that we can think about. Now, what's useful about that is that oftentimes those categories, although there may be many individual ways that one or the other category can get acted, there are three categories. And oftentimes what that means is there may be coalescence. So it's not like there's 100 different ways and you're going to have to go target 100 different things. They may converge on a few things. And so what you really need to do is come up with a combination against those few things. So that's a hopeful, and I think actually it has the benefit of being true in some cases. So the rational basis for developing therapeutic combinations in large part may have to do with understanding, is there sort of a final common pathway or one or two or three final common pathways of how all resistance mechanisms go? And if so, you hit those and you're done. Now the other way you can deal with it is say, well, there are other pathways that cancer cells depend on besides the ones that are mutated by these driver oncogene events. So maybe you just also attack those side by side. That's sort of how HIV therapy works. I mean, there are multiple viral processes that are required, and you just target 3 or 4 of them all at once, and the virus just can't overcome that. So those are the two, at least medical ways that we think about it. And the third way that we could go after it is, as I mentioned before, if we can harness the immune system, that would be another way to do it. So there's definitely hope for overcoming resistance, and it's a major area, a very exciting area right now.
My question is that it seems that as people age, they tend to have more opportunities for cancer. With your examination of the telomeres, as it gets older, more damaged, more frayed, there's more opportunities for problems to arise. In your investigation of cancer, are you finding insights that might imply new understanding of how people age and how we could potentially address the damaged or frayed telomeres?
Yeah, it's very interesting because normally what happens in normal cancer cells, normal cells, not cancer cells, in normal cells, they divide, and the telomeres, the ends of the chromosomes, they get shorter, and eventually they get so short that the cell can't divide anymore. And what happens is it's called senescence. It just sort of freezes. It's not dead, but it can't divide anymore. And so one of the theories about how aging occurs is that over time, the proportion of your normal cells that are in senescence in various vital organs, in the skin, et cetera, is growing, and that may be sort of part of the aging process. So ironically, the cancer reverses that process in the wrong cell. So in essence, in that regard, cancer is sort of a it's an aberrant, perverse counter-aging mechanism.
So we're coming on the hour, and I want to invite any students in the audience who has a question to raise their hand so that we can have the last question from a student. You can stand up because you're...
So are you able to determine whether the cancer on a specific individual was caused by a virus or whether it was caused because of genetic mutations?
Great question. Very often the answer is yes. So there are some cancers that we know are very commonly caused by viruses. So for example, cancer of the head and neck is commonly caused by a virus. Some cancers of the liver are caused by different viruses. So sometimes you just know based on, you don't even have to look at the tumor, you can take a history of where the patient lived or what their habits were, et cetera, and you can determine whether a virus was a likely cause. But even if you don't do that, as long as you have the help of a computer algorithm that tells you to go look for all the viruses in the world, if you sequence a genome, you will know if there's a virus in there. So it's interesting, one of the projects that we did in the lab a few years ago was actually to do a genome sequencing project of head and neck cancer. And just by sequencing these genomes and not doing anything fancy, we were able to identify exactly the subset of head and neck cancers that were caused by human papillomavirus and which subset of human papillomavirus caused it. We were actually better than the standard of care in identifying that. So it can be done, and it really but one has to sort of have the right computational tools to do that. So the short answer is yes.
Okay. Unfortunately, we're out of time for questions here, but I do want to remind you that Levi is going to stay for a while. We have a reception, so please continue the conversation. He'll be happy to answer some questions informally and with each other. Thank you again for coming. I can see from the number of questions that we're likely to have a very full crowd next week again, Levi.